zat er iets simpeler.
Ja, want het was voor jonge
Heel,
met zwarte
ja,
tekst.
ja.
En volgens mij niet heel veel meer. Want ik heb nog niet
Het was klein, kleiner boek.
zo'n boekje zo, ja. En volgens mij ligt het nog boven de kist.
Het is allemaal veel te technisch voor ons. Jessica, gaat dit eigenlijk wel over die apparatuur die... Over die apparatuur die voor de astmafonds of voor de astma?
Herewith, I open this academic session in which the candidate, Mr. Stavropoulos, is going to defend his thesis entitled Exhaled Breath Analysis, the Road Towards Its Clinical Implementation. I welcome you, of course, and wish you lots of success together with your partners. I also welcome your supervisors, Professor von Schoten and Professor Smolinska. Obviously, welcome to the opposition, online and on site, and the audience here and the audience online. But before we start the opposition, I give you the opportunity to present the data that you have presented in your thesis.
Dear Prorector, dear committee members, dear family and friends, dear audience, thank you for being here today. For the next 15 minutes, I'll be walking you through the main findings and examine topics of my PhD thesis entitled Exhaled Breath Analysis, the Road Towards Its Clinical Implementation. Breath analysis has been examined since the ancient times. For example, the Greek physician Hippocrates was the first one to notice the characteristic breath odor of people that suffer from liver diseases, which he called fetor hepaticus. Now, in 1989, the first dog sniffing out cancer report was published, where it was basically reported that the dogs can smell cancer. And even more recently, in 2019, another report was published where basically a woman was able to smell Parkinson's disease in her husband. So all this means that there must be something in exhaled breath or other bodily excretion means that if captured, it can probably help us diagnose diseases. Now that something is what we call volatile organic compounds, or shortly VOCs. An exhaled breath sample consists of a variety of gases. You can think of oxygen, nitrogen, carbon dioxide, some noble gases, as well as the volatile organic compounds. VOCs are low molecular weight molecules that exhibit a high vapor pressure at room temperature. Now, VOCs can originate either endogenously, and here you can think, for example, an inflammation of a human organ within the body. That's what endogenously means. Where what happens then is that those VOCs are released in the bloodstream, the human bloodstream, and then eventually excreted via breath or other bodily excretion means. Those VOCs can also originate exogenously. so from outside the body. And here you can think, for instance, the environment, someone's diet, or even lifestyle. A good example here is that to think of the smell of people that smoke or drink alcohol. Now, exhaled breath has not been implemented in the clinics yet as a standardized way for diagnosing and monitoring diseases. Therefore, in my thesis, we wanted to examine what might be hindering this. And more specifically, we wanted to study technical aspects of exhaled breath data. So basically, how one should go about conducting an exhaled breath VOC data analysis. At the same time, we also wanted to perform our own case study, where we would examine primary sclerosing cholangitis, which is a liver disease impairment. And in doing so, we also conducted a literature review where we wanted to see what has been reported so far in terms of VOCs in liver impairment. Now, first, we wanted to compare results and data from different studies, and that led us to examine what we call batch effects. Now, batch effects are unwanted variations or information embedded in the data, which can originate either from methodological or environmental sources. So, for example, you can think here the way we sample a sample, the way we analyze the sample, or the way we handle the data of that sample. Now, ideally, to eliminate those batch effects as much as possible, one would have to sample, measure, and analyze everything at the same time, at the same location, by the same personnel, something which is obviously not feasible. Now, if we think, for instance, for example, to understand what those batch effects are, think, for example, a cohort that we want to sample their breath. And then we sample their breath in two different time points. So what you see here on the screen are individuals that provided exhaled breath samples, and we took their samples twice, time point 1, the blue data points, time point 2, the yellow data points. Now, because those samples in those two different data points came from the same individuals, so that means that the biological information is the same, we should be able to see an overlap between those data points and not this distinct separation. So that basically means that we have batch effects in the data that we need to correct for. And if we were to correct for those, we should get something like this. So basically here you see that the data points or the measurements, the breath measurements overlap, and this external variation, non-biological variation in the data has been removed. So first, we wanted to compare and investigate 3 different data sets coming from 3 different diseases, in particular inflammatory bowel disease, irritable bowel syndrome, and chronic liver disease. And the goal was to see if we can find biomarkers, VOCs, that can help us differentiate these groups of people. However, those diseases share a number of symptoms and also some pathophysiological mechanisms. So that means that if we were to visualize the breath samples of those patients, we should see in a 3D figure, as you see here on the screen, these data points represent those patients, we should see some sort of an overlap amongst these data points. And as you see here, that's clearly not the case. You see that the IBD individuals are clustered alone in the red, dark blue, the IBS, and light blue, the CLD individuals. So that basically led us to believe that we have batch effects in our data. Now, we wanted to correct for those, and to correct for those, we applied what we call batch effect correction algorithms. Now, these algorithms have been developed to correct for batch effects within the same study, within study batch effects. So you can think here batch effects only on the IBD dataset, not across study batch effects, which was basically what we wanted to do. But nonetheless, we applied those algorithms because they haven't been developed so far across study batch effect correction algorithms, and wanted to see what we could get. And by applying those, this is what we got. So you see here that instead of removing these batch effects, the unwanted information in the data, instead we got an even greater separation amongst the 3 data sets, which basically led us to believe that the batch effects are still there. And the fact that the data points per dataset have shrunk basically meant that we maybe have removed biological information instead, which was what we wanted to keep. Next, and as part of our case study on primary sclerosing cholangitis, the PSC case study, we conducted a literature review on VOCs in liver impairment. And what we found was that several studies have published several VOCs that seem to have a solid biological origin explanation. And what we did, what you see here also in the figure, was that we summarized all those metabolic pathways that have been reported for those VOC compounds. So for example, if we take limonene, as you see here, what happens with limonene is that limonene is normally metabolized into peryl alcohol or transcarveol, and then it's excreted via the urine. However, when the liver is diseased, we don't have this conversion of limonene to PA and TC. Instead, limonene is stored in the adipose tissue of the human body. Therefore, it accumulates in the body, and therefore we have the excretion of limonene in the exhaled breath. And here is a table of all the VOCs that have been reported in the literature in terms of VOCs in liver impairment in more than one study. Now, what we found was that the second most reported compound was, in fact, limonene. And that was surprising because limonene is an exogenous VOC. It comes from outside of the body. It's not an endogenous VOC. And in fact, it comes from foods such as citrus. Now, by taking all those VOCs into account, we performed our case study on primary sclerosing cholangitis. Now, PSC is a chronic liver disease that can cause multiple stenosis and segmental dilatations of the bile duct. PSC can lead to liver failure, malignancy, and/or death. Now, on the left-hand side figure, you see where cholangitis is located in the human body, in this small circle. Whereas in the middle figure, you see how the bile duct looks like when it's healthy. Whereas on the right-hand side figure, you see how the bile duct looks like when it's affected by PSC. Moreover, 80% of the PSC-diseased individuals also suffer from IBD. However, the opposite does not necessarily hold true, since only 5% of the IBD-diseased individuals also suffer from PSC. Now, on top of that, PSC-diseased individuals that also suffer from IBD show mild to no IBD-related symptoms, and thus that makes the PSC diagnosis rather challenging. So the aim of the project here was to see if we can find VOC biomarkers that can help us discriminate PSC-diseased individuals, whether they had IBD or not, from IBD-only suffering individuals. And so here are the results of our study. So basically, the prediction model that we used gave us a sensitivity of 77% and a specificity of 83%, or in other words, 77% certainty that someone suffers from PSC, either with IBD or not, and 83% certainty that someone suffers from IBD only. And if we were to visualize those patients, the breath samples, what you see here in the figure, you see that the PSC-diseased individuals are clustered mostly on the left-hand side of the figure, whereas the IBD-only diseased individuals are mostly clustered on the right-hand side of the figure. Furthermore, except for sampling breath for those PSC-diseased individuals for our PSC case study, we also sampled feces, and that was based on the leaky gut theory. That theory hypothesizes that there must be an ongoing inflammatory stimulus that originates in the gut and that supports the inflammation in the PSC cases. Therefore, there should be some detectable compounds in both breath and feces. And what we did then was that we wanted to see whether if we combine, or as we say, fuse breath and fecal data, if we can get a better understanding of the PSC etiology and pathogenesis. So we developed what we called a proximity stacking data fusion tool. And how briefly this tool works, if we assume here that the yellow table represents the breath data, whereas the purple table represents the fecal data, then what happens is that these data sets are stacked one on top of each other, hence the name stacking. So instead of using two data sets, we used in the end one for our further analysis / classification. And so in order to test the performance of that data fusion tool, we sampled Crohn's disease individuals that they were either in a remission state of the disease or they were active cases of the disease. And more specifically, we sampled breath, feces, and blood. And we wanted to fuse all these three different data sets to see how the data fusion tool would perform. And so by using this tool and the prediction model, we achieved a sensitivity of 93% and a specificity of 100%. Or in other words, 93% certainty that someone is a CD active case and 100% certainty if someone is a CD remission case. And if again we were to visualize those patients and their breath samples and the fecal, combined with the fecal and the blood samples, you see that the individuals that were in a remission state and they are color-coded blue, those data points, they are nicely clustered on the left-hand side of the figure, whereas the individuals that were active cases of Crohn's disease are nicely clustered on the right-hand side of the figure. In conclusion, we raised awareness that batch effects arise in breath data, and we need to pay special attention not only when we handle the data, but also when we sample and analyze the samples, because it seems that currently correcting for external variations in the data sets while we handle the data is challenging so far. Then next, and via our literature review, we reported that the second most reported compound seems to be an exogenous compound, which is limonene. And that led us to suggest that maybe the breath research should also focus on performing what we called exogenous VOC analysis, where in theory, someone would expose a cohort of individuals to a certain concentration of a compound, say limonene, in order to be metabolized by the liver, then sample their breath after the exposure and then check the concentration between the inhaled and exhaled concentration of that compound by the individuals. And by checking those differences in concentrations, we might be able to say something about liver function. Then next, we also showed the potential of applying VOC analysis in classifying PSC diseased individuals versus IBD only suffering individuals. And lastly, we developed what we call a proximity stacking fusion tool, which can be used not only in breath research, but in other data science research applications as well. So thank you for your attention, and now I would like to give the floor back to the prorector.
Thank you, Mr. Candidate, for a nice lecture. We will now start the opposition, and the first opponent is also a member of the assessment committee, and it's Dr. Ciacenti. And he's professor in data analysis and modeling models at Wageningen University. And thank you for coming all the way from Wageningen. The University of Maastricht is always glad to have people from outside. The floor is yours.
Many thanks, and dear candidate, first of all, congratulations for the completion of your thesis, which I've read with great pleasure, and also congratulations to your supervisor. And I would like to start from data fusion, which is a topic which is pretty close to my interest. And in particular, I would like to ask a question on a sentence that you write on page 67, which took me a bit by surprise. When you write that low-level fusion is the simplest of all data fusion approaches. So I would like to know why you said that is the simpler, because in my opinion, it's actually the most complicated.
Esteemed opponent, thank you for your nice words and for the question. In my opinion, low-level fusion is the simplest because we just fuse the data as they are. We take the entire data sets and we concatenate them next to each other. Whereas if you have to think about mid-level, high-level, or even the one that we proposed, there we have to, for mid-level case, we have to find significant variables or features if we perform some PCA analysis or PLS analysis that needs to be fused from per data set. That's something, these might be challenging sometimes in terms of scaling of the data sets, because we need to scale those data first before we perform this sort of analysis. In terms of high-level fusion, that might also be, it's a bit more challenging in the sense that we have to perform classification analysis, get the results from multiple models, and then we need to fuse them, and we have to also see which method we need to use, either that is majority voting, for instance, or adjusted probabilities, as we did in our case. And if we also talk about the other more advanced cases, either the one that we proposed or other kernel fusion approaches that have been proposed so far, that gets a bit more complicated in the sense that if you talk about kernels, then you have to find the right loss function to get those kernels, and then also find how you're going to fuse them via weighted sum, how you're going to go about this. In our case, with the proximities, you also need to do some sort of analysis before you go about this. You also have to find the weighted sum, or the linear way you want to fuse those data sets. So yeah, in that regard, I think low level, even though these, I forgot to mention that these also required some scaling when we want to fuse the variables, of course, but it seems the one that has the least work to be done in order to fuse the data set and go about our analysis.
Okay, many thanks for this articulated answer. And if you're allowed, I will like to move to a question about your proposition. So I would like to ask one of your paranymph to read the proposition number 6.
The omics field has had a significant impact on many areas of biology and medicine. It remains to be seen whether volatile omics could be added to the omics field, and if so, what impact it could make. Okay,
given that your PhD work is about exploring different aspects of volatiles, so what do you think? Is a go or a no-go, the volatomics, and why?
So I think there is definitely something to be found in volatile data. It's apparent from all those studies, and also since the breath has been used since ancient times, there is something there, and also with those clear examples of dogs or this lady that could spell Parkinson. There is something in there. But so far it's challenging to capture that consistently and also reproduce those results. So in my opinion, there is definitely future in volatile omics data. It will be implemented in one way or the other in this whole omics field. However, I believe it is still going to take time. A lot more research is needed. There are still a lot of aspects, in my opinion, that need to be dealt with before we can say that we can perform volatile omics analysis properly. So all in all, I believe, yes, there is future, but it's still going to take some time before we manage to implement volatile omics in its entire omics field.
Many thanks. I think there is no time for another question. Right?
Thank you. Thank you for your opposition, Dr. Sargenti. Then we come to the following opponent, who's also a member of the assessment committee, and that's Professor Davis. And she's Associate Vice Chancellor for the Interdisciplinary Research and Strategy Initiatives in the University of California. And thank you, Professor Davis, for coming all the way, albeit online. And I appreciate it because it's also only 7 o'clock in the morning in your area. So the floor is yours.
Wonderful. I'd like to add my congratulations to the candidate and his advisors as well for completing the study. And I'm looking forward to hearing more about the work. You mentioned confounding effects as something that you were definitely interested in your work. And the batch effects that you found from your time points, that was significantly interesting. And it's something that we struggle with in the field. Do you have any other ideas about what other confounding effects might be significant? And if you were to give advice to a new study starting, what else would you tell people to look for?
Highly esteemed opponent, thank you for the nice words and your question. So the question is, what other factors need to be considered, or I'm not fully sure I understood the question. If you could please repeat it.
Absolutely. So you rightly pointed out that batch effects from time sampling are one significant confounding factor. What other confounding factors should you be aware of?
Yeah, definitely from a time standpoint, that's it seems to be a major factor. I would also definitely add the environment, the analytical tools that are also used. For instance, in my case here, we used GCMS, but from our literature review, we also saw multiple studies using ShiftMS or GCMS or IMS, all those MS-based approaches, or even ENOS, but that's a different technique altogether. Personnel, definitely. We've also noticed that even though the location or the time might be different, personnel conducting the analysis can also introduce non-biological information in the data. So yeah, I definitely believe that it's not only the time point, is the analytical technique that is used, the personnel, and most definitely the environment and the diet as well. The diet, I believe, also contributes a lot to seeing external variations in the breath samples.
Great, thank you so much. And then as you were exploring the confounding effects that you did see so far, do you think that there's more than one way that you can correct for these? You talked about the stacking methods, but what else can you do to correct your data?
You mentioned which mentions? I said I didn't get the word.
You were talking about data stacking, I think, earlier. And what other effects, what other approaches could you use to remove the confounding effects?
So, yeah, as I said, ideally you want to sample everything at the same time or by the same personnel or location, which is obviously not possible. What we proposed in our own case study, and also based on the literature and metabolomic studies, is to use quality controls. And at the same time, we might also could use an internal standard, inject an internal standard in the samples as well as in the quality controls. Now, by using those QCs, we can see whether we have external variations, because the QCs are known, it's the same sample. So if we analyze those in regular intervals throughout the analysis, they should be the same. So if we see differences in there, that means that something is wrong in there. Also, by injecting the internal standard in the samples, we can monitor whether there have been changes in those samples. However, these might also be tricky, because we need to be very careful with the internal standard that we use, because it's not like we can inject any compound and go about it, because this compound might interact with the sample. So this is also a bit challenging, and we need to pay particular attention to it. But so far, from what we've seen, I would say that the way to go about it is to use quality controls to ensure a high quality analysis, and also the use of internal standards in our samples, and of course the quality controls.
Wonderful, thank you. That's all of my questions.
Thank you, Professor Davis, for your opposition. The following opponent is Professor Faucon. He is a professor in analytical chemistry in the University of Liège, and that is in Belgium. And thank you for coming all the way from Belgium as well.
Thank you, and congratulations for the thesis, the work, to you and the supervisors. You know how much I'm, interested in that topic, and I really enjoy the reading. Very well written, very easy to get into it, and very exciting to turn the next pages. So, good job for that. I've got a very general question, because you've been mentioning data fusion, because you could go for, let's say, hybrid metabolomics, considering like different matrices like blood, breath, feces, urine, etc. You could also use different instrumentation, GC mass spec, PTRMS, SIFT-MS, e-noses. You could also use endogenous, or you also suggested, very interestingly, to use exogenous compounds. So it's so many possibilities, How would you narrow down the best one? As of today, what would be your guess to go to get the best chances to be very specific to a given disease? What would you do?
Highly esteemed opponent, thank you for the nice words and your question. In terms of instrumentation, what you mentioned, I've only explored GCMS, so perhaps if I had to redo it again, I would go, I guess, for the same. However, I have to say, GCMS might also be a very strong possibility for us in this case, and this is because the VOCs in the breath samples, we see a lot of times that there is coelution of those compounds in the retention time, and therefore by using GCMS, we might achieve a higher separation. However, that also comes at another cost, of course, of using that technique. So far in the literature also, GCMS is the predominant technique of choice, whereas GCMS is used for other types of compounds, I believe, maybe like fatty acids or this sort of compounds. So between these two, probably I would go for GCMS still. However, I'm not entirely convinced whether if we do a breath analysis, we should go for MS-based techniques altogether, in the sense whether we really want to go for compound identification. And that's what the MS-based techniques do. GCMS, they identify the compounds, whereas one maybe could use e-nose, where we'll get a breath profile. So I believe if I had to reduce something related to breath, and then if it was about diagnosis of a disease, I would maybe go for a breath profile like the e-nose instead. However, if we talk about understanding the disease itself, pathophysiological mechanisms and compounds relate to the cause of those diseases, I would really be interested in identification of the compounds, and therefore I would go for an MS-based technique.
And to maybe bounce on what you just said, how would you then introduce the sample to the e-nose that you would use? Would you just ask the patient to blow into the device, or would you use a specific trapping device? What would be the idea?
As far as I know, with the e-nose, the patients just blow in or exhale in the e-nose device. Whereas if we want to go about the MS-based techniques, then have to provide them these like a mask or a bag where they exhale, and then we have to take those samples, analyze them via a mesh-based technique, and then go for
because that's exactly where I want to drive you, is that you did everything on GC mass spec using trapping devices. And I think just because, and we do the same, I'm not criticizing here, but because of that, you introduce a selectivity in your entire process. A lot of compounds will not be retained on the TD sorbent, for example, right? And then if you shift from there, where you developed what you did, to a non-selective trapping device where you feed your analytical device with everything that's getting out of your patient, you might have very different outcomes. I'm not saying that it will be the case, but I think that's something people maybe underestimate a little bit in terms of trapping. But I think it's something to maybe discuss later. But in terms of social security, because you want to move to the hospital. So always the question when you move to a hospital is how much does it cost? Because the social security has to pay at the end, right? So do you think you're going to be nicely contributing to saving everyone's money by introducing such technique in hospital or not? What's your idea about that?
Yeah, so I did a little bit of research on that, but based on statistics that have been published in terms of American diseased individuals and the healthcare system, and especially for gastrointestinal diseases. I think they reported back in 2018 or 19 a few hundreds of billions spent on healthcare costs with the current techniques. Those techniques, for example, include biopsy or imaging, expensive techniques. And for that, as you say, the health care system or the insurances need to pay. I believe that if we were to implement breath, we can significantly reduce those costs because, and also make it a lot more patient-friendly, but a reduction of cost in the terms that you will have probably a simple device where you exhale and then you get your results, which can, and you also get on real-time results. So basically that means you won't have to have as much personnel probably to conduct the rest of the analysis, so you can also save some costs there as well. So yeah, I strongly believe that implementing breath can significantly reduce the costs in the healthcare system, not only the States, of course, but worldwide as well.
Okay, thank you.
Thank you, Professor Faucon, for your opposition. And the following opponent is Professor Jonkers. She is Professor in Intestinal Health and Scientific Director of the Institute of Nutrition in the University of Maastricht. And for today, she has also accepted the role of Secretary of this committee, for which I'm very grateful. The floor is yours.
Thank you very much. Dear candidate, I would like to congratulate and compliment you and your supervisory team. You had a very nice thesis. I think it's really important work. 6 chapters, 5 of which already published, so compliments for that. And I think it's a very important topic nowadays to get further insight on how we have to deal with those big data sets which we all are collecting. And I really believe that there's a lot in the breath samples. But I also would like to follow up a little bit on my previous opponent to try to understand how we can bring it to the clinic. And you already indicated the road towards its clinical implementation. So let's see where we stand. And I was triggered by several chapters, but let's just start at chapter 2, where you actually described a lot of nice compounds which have been identified and associated with liver disease. And although I'm very convinced that there's a lot of information in there, I struggle also to understand how disease-specific it is. Because if we take 2 examples, for example, you have the alkanes, which is related to lipid peroxidation, oxidative stress, inflammation. It's associated with, it's more pathophysiological mechanism associated with several diseases. You had propionic acid, for example, which you link to the liver function, but I think can it not just simply be due to diet and microbiome? So What do you think? How disease-specific are your profiles, or are they more specific for certain pathophysiological mechanisms? What is your opinion?
Highly esteemed opponent, thank you for the nice words and your question. I believe that those compounds that have been reported so far are mostly symptom related instead of like disease related, because what has been examined so far is basically to find VOCs regarding the symptoms of our disease instead of what has been the cause of the disease. Also, as you mentioned, we have alkanes that have been allocated or associated with a number of different diseases. So yes, it's possible that they are all present in those diseases, but on the other hand, how disease-specific they can be. Yeah, as I said, in my opinion, they are not fully disease-specific, and for that, we need to do a lot more work on it. I would say that the way those studies have been done is to get maybe an idea of what compounds might be present there. And what we should do perhaps next is to go for some in vitro analysis or animal studies where we would specifically try to find those compounds and see whether they are disease specific or maybe they originate from dietary interventions or medication. And of course, that also is a bit challenging, because then you have to define the VOC compound origin, structure, and so that requires also a lot more work. But as soon as we do that, and then we conduct those in vitro studies or animal studies, and then we confirm that this VOC is produced in human diseased individuals, then we can go about it and then just perform a targeted approach targeting this particular VOC, and then we can say with certainty if it's disease-related or not.
No, I totally understand and agree that we still have to do some follow-up work. I was also triggered with the fact that you mentioned diet, of course. You already also mentioned it by the previous speaker, because I struggle a little bit how to interpret diet in the context of VOC. Should we consider it a confounder, or should we also take it into account as a risk factor, because it's really also often related to diseases? So how do you see that and how to deal with that?
Yeah, diet definitely plays a role in it. I think mostly is considered as a like confounding factor so far, that it actually influences our results and the VOCs that we find. However, I do believe maybe we can still use it somehow, like to connect VOCs with dietary interventions. So what I mean by that is that perhaps we can find foods that trigger the release of some certain VOCs for one particular disease.
And do you think that we really have to check for the composition of diet? If we measure patients, is it important, or can we just ignore it?
No, it's absolutely important. So if I understand it correctly, you mean if we want to sample breath, whether we should take diet into account as like confounding factor, or take care to prevent that from happening? Because a nice example here would be that, yeah, if we eat garlic, then breath smells. So it means that we should not just maybe fast before we sample breath. So in that regard, yes, it really can influence our samples. But then again, maybe we can also use those dietary interventions for different types of analysis, where, as I said, we can find specific compounds that are triggered by specific foods, and then we can measure those, and then we can see whether this dietary intervention for IBD or IBS patients in particular works or has the effect that we want it to have.
That's very interesting to learn. Can I ask a small final question? Yes, I still have to tell. Because of course, if you talk about diet, and you already mentioned pathophysiological processes that we have to learn from how they are involved in diseases. But then there's another factor to take into account, because I also noted in a lot of your metabolites, like alcohol, acetaldehyde, I'm biased, I directly consider then the microbiome. So is it not just microbiome composition what you're measuring, or is it really disease or pathophysiology?
Yeah, that's a good question. However, I believe it might be disease specific. What I mean by that is if we consider IBS, for instance, or IBD, because we have GI tract symptoms there, I believe there is information not only in feces but also in breath. So in that case, we can have complementary data. However, if we were to examine another disease like a lung diseases, then I wouldn't think of gut microbiome in that case, or if we measure something that has actually come from that came from.
Don't underscore the gut microbiome in extraintestinal diseases.
Yes. But I think it might be like more disease specific. It depends on the disease that we are looking at, what we have, the question that we have, the research question that we have at hand. I think it depends on this. But indeed, gut microbiome is a good one. Yeah, definitely. We might, and we also saw that with our studies when we tried to find compounds in gut microbiome for Crohn's disease, how well it actually performed with our analysis. So,
and to make your analysis more disease specific, should we more often consider adding specific samples, like if we talk about intestinal diseases, features, and if there are other diseases, it can be saliva samples or whatever. Do you think that can bring us a step closer to have disease-specific markers?
So you mean if we combine multiple means?
If we think about what kind of disease you're looking for and combine then the specific biosamples which better reflect a certain organ or so, could that be a step which could be helpful?
So I think that relates to the fusion case that we did, which was on Crohn's disease. Because it's a GI tract disease, and I said that I believe we can find compounds in both breath and feces. So in that regard, we can get multiple these two data sets, combine them, and perhaps we can get a better understanding, or like in the PSC case, a better understanding of that disease. Now, I believe it's based on the disease that depends on what we have. It might be really beneficial to combine multiple samples.
Okay, thank you very much. I'm very happy with your answers, and give the floor back to the pro-rector.
Thank you very much for your opposition, Professor Jonkers. Now we come to the following opponent, and that is Dr. Cavil, and she's assistant professor at the Department of Advanced Computing Sciences in the University of Maastricht. Dr. Cavil.
Thank you, Pro-Rector, and dearest candidate, thank you for the opportunity to read this interesting thesis with many interesting and also new to me techniques for data integration, for dealing with batch effects. I'd like to zoom in a bit to start with on chapter 6, and in particular page 152. In the top left on that page, you show scores plot in figure 5. And in my experience, normally when we're doing machine learning, we want our training and validation set to be as similar as possible to each other. The validation set, the training set is representative of the population that you're looking at. But here, particularly in the active, so we have the training and validation set for the remission and the active samples. And in the active samples, I really see quite a separation between the training active and the validation active. Training active shown in red and the validation active shown in green. I was wondering if you could reflect a little bit on how this figure was made and what this separation might mean. Do we have any explanation for why our training and validation set look different here, or is it still unknown?
Esteemed opponent, thank you for the nice words and the question. You spoke about figure 5, if I'm not mistaken, and the red
figure
data
5,
points.
top left.
And and why we see these variation in the data points. That was
yeah, particularly in the active set. So
Yeah.
the training set and the validation set seem to be quite separate there in that one class.
If I had to speculate on something on this, why we might be seeing this variation in the training, is that we used here the unsupervised random forest proximities, and this one does not take the class into account. So that might be one of the reasons why we don't see such a nice grouping of those groups.
In the process of generating this plot, is the unsupervised random forest just applied to the training set, and then the validation set is projected into the unsupervised random forest?
Could you please repeat, because I didn't hear you correctly?
Sorry. So when you make this plot, are you building the unsupervised random forest on all the samples or just on the training set, and then projecting the validation set in?
Yeah, that's correct. We used all the training samples. We got the proximities via unsupervised random forest, and then we projected the validation samples.
So might we be looking at some overfitting here then, if the validation samples don't look like the training, an overfitting of the training set, do you think? Maybe.
I think that overfitting is always possible. However, I would say that in case of unsupervised random forest or random forest, we eliminate as much of it as possible, overfitting, because we have the trees that are built in parallel instead of sequential. In theory, you may also argue the more trees, the merrier the result. And even though you can still have some overfitting, because in the end you average them out, all those trees, this overfitting might be reduced. So again, I think there is a chance we might be seeing some overfitting in there, but I don't think so that it's that high.
Okay, thank you. I'd like to zoom a bit further out now, and in your general discussion at the end, on page 189, you defined high-quality data, and I've lost it, but I wrote some notes. It said that high-quality data had no background noise, no biological variation or instrumental artifacts. And I agree, if I had data that had no background noise, no biological variation or instrumental artifacts, it would be very high quality. But is that really realistic for our data to have none of those things?
No, no, this is really not possible. No.
Yeah, and so I guess I want to ask what features do our algorithms then need to have to deal with perhaps this non-high quality data that we're going to be dealing with?
So what basically, what an ideal algorithm should do in order to deal with those problems that we might, the quality problems that we might have.
Or perhaps workflow as a whole rather than specific algorithm.
Yeah, yeah. So in our case here, I think most of it starts before the data analysis and the handling and the choice of those algorithms. I believe that we should really pay close attention to how we sample and how we analyze, because all those features, as we say, those bad features that are introduced in the data might come from there. And those algorithms, there is only so much they can do. So primarily I would focus on doing my best at the beginning to prevent all this from happening and then help get some high quality data. Now, of course, if we take, for example, the breath data, we have maybe some non-linearities in the data. So basically that means that we should also have like a good algorithm that basically can read those non-linearities. So maybe that's something like a feature of an algorithm to be used. Yeah, something like that, I think.
Okay, I'll pass the word back to the prorector.
Thank you, Dr. Kavil, for your opposition. The following opponent is Dr. Gottschalk, and his expertise is genetic toxicology, and in that role he's associate professor of the Nutrium Institute of the University of Maastricht. Dr. Gottschalk.
Thank you, Prorector. Dear candidate, first of all, congratulations with your nice booklet. It's not only the content, but I think also how it looks. It's really nice, and I would like to congratulate that. I would like to congratulate you with that as well, and I would also like to congratulate the promotion team for this result. I would like to continue actually on the discussion that you had with Professor Jonkers on the clinical application of breath analysis, because I think we are far from there, right? Because volatiles in the breath can be considered as a biomarker, and unfortunately, quite a lot of biomarkers are being implemented in the hospital and then being retracted because they do not do what they promise to do. Although they may be promising, they are retracted because simply we do not understand them well enough before we apply them. So I think we need to understand the biomarkers better before we start applying them in the clinic. And that is actually the discussion that I would like to do with you, to have with you. One of the factors that we need to understand is intra-individual variation. So not inter, but intra-individual variation. If a patient comes at the doctor, so you let him analyze, you analyze his or her breath, and it is a patient has a certain disease. You want that patient also to be diagnosed as a patient next week. Or if he would come in 2 weeks, 1 month, half a year, it's still a patient, right? But if you come in half a year, it's a different season. You are having winter versus summer, lifestyle is different in winter versus summer. So you do not only have batch effects because you have to measure at a different time point, you also have biological reasons of batch effects. So do you think that we will manage to have breath analysis at all in the clinic if you take all those factors into account? Because it will be very complex if those batch effects are there.
Esteemed opponent, thank you for the nice words and the question. Indeed, this is an interesting topic to see whether breath will be ever implemented because of all those external variations that we have. Now, what I personally believe is that, as we also discussed earlier, when it comes to diseases, we should focus on specific compounds that requires a lot of work, either in vitro or animal studies to find that compound. And if we do all that work, in theory, yes, that means that this compound is there. So it won't be dependent on like external variations. And what we could possibly do in that case is if to go about doing an exogenous VOC analysis. So say we identify a particular compound that's related to, I don't know, IBD, then exogenously we can provide to the patients, and then we can see, as we said, with the different concentrations, what has happened throughout these months, and maybe identify or diagnose the disease.
So then you would identify compounds that are not affected, for instance, by season or by changes in lifestyle or whatever. So you want compounds that are more stable.
Yes, exactly. Because if it's something exogenous that we can provide it in the form of a pill, then it won't be affected by external factors, whether it's summer, whether it's winter, or the diet, for instance. It will be just what it is on its own. So that's one way to go about it. And I think what's been happening so far, or what we might end up doing, because this is also very challenging, is to use breath in the clinics as a screening tool first, like the upcoming years or so, you can find such an application where we basically get an idea of whether that individual suffers from that disease or not, and then we can proceed with further other diagnostic tools. Because if we think, for example, diseases that require that require a biopsy, we could maybe first, and biopsy is expensive and invasive, we could perhaps first implement a breath test. Say you have a 70-80% certainty that that person might have that, then you can send that person for the biopsy. But if it's not, then you can just skip from sending that person to biopsy, and then that way it will be definitely beneficial not only for the person, but also, as we also discussed earlier, for the healthcare.
But would that not be a pity to only use it as screening? You discussed with Professor Jonkers that specific compounds may be really disease-related. So if you intervene in the disease, so you treat the patient, maybe you can also use that compound to see whether the disease disappears, right? Because you can follow up the changes in that disease specific compound. So wouldn't you not also use it, for instance, for prognosis?
Yeah, absolutely. Ideally, that's what we would like to do, to establish breath tests, nice and simple, where we just diagnose or say, okay, now you're cured. But because it's influenced by so many factors, and it at the moment seems challenging to define with high certainty, and actually develop with a breath test with higher certainty whether someone is diseased or not. So for the foreseeable future, the way I see it is that it will be used as a screening tool, whereas maybe later with further research we can perhaps have a definite diagnostic or monitoring tool.
So you may finish, or you were finished?
I was finished.
You was finished. Good. Good. Thank you, Dr. Godschalk, for your opposition. And Mr. Stavropoulos, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I request you and your company online and on site, here present, await the results of our deliberations and our return in this room. And with that, I suspend the meeting.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. The mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. Better future that we go for. And Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
not supposed to say now.
I reopen this academic session. Mr. Stavropoulos, the committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor van Schoten is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Georgios Stavropoulos, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Someone a photo camera. Okay.
It's yours now. Dr. Stavropoulos, dear George, congratulations. I'm very proud of you. And the honor is to your supervisor, Aki Smolinska, to praise you in the laudatio.
Thank you. Dear Dr. Stavropoulos, I hope I pronounce it properly. Please accept my warmest congratulations. Dear George, it is my honor to be, well, second one, not the first one, to say I'm so delighted to know that you have already reached your goal. You truly have proved your self-dedication and your work. It is with great honor and pleasure that I stand before you to deliver this laudatio, paying tribute to your dedication, intellect, and scholarly contribution. I want to personally congratulate you for being such a great success and setting a good example for other colleagues. Together with Professor van Schoten, your promoter, we are proud that you have gained your PhD degree after all those years of hard work and hardship in the department. Remember your study, data, the thesis. Every time you were thinking that you were almost there, but you often did not see the end, right? Wow, look at you now. This all has paid off. Again, congratulations. Through your research, George, you have delved into uncharted territories, expanding the knowledge in the field of chemometrics, but also exhale breath. Your work has not only contributed to the existing body of the literature, but has also sparked new avenues of inquiry and opened doors to fresh perspective. We know each other for more than 5 years. So for the next few minutes, I want to remind the time that you spent in the department. You joined the department in 2017 in September after completing your master internship at Maastricht University and your internship at AkzoNobel. I remember the first day you came to us for the job interview. Then you came to present your awesome work that you did at AkzoNobel. And I remember how you were just like, yeah, so confident and zero stress. At that moment, I just felt like, wow, you are such an easygoing, confident, and relaxed person. You start at the department was rather challenging because me, after 2 months, I just decided to go on maternity leave. So you were left literally, well, not alone, you were left with Professor Van Schoten, but it was difficult because not only you had to dive into Excel Brev, chemometrics, but I made it even more difficult because I asked you to take care of my course and you had to teach on Excel Brev and also practical in machine learning and using a problem-based learning technique that is completely, it was completely new to you. But you seem to get very good in that. You seem to be very natural with the students and you seem to have very good connection with them. So, the start of your PhD, I think you were put into really deep water, but as we can see now, you have showed how well you can swim. In the past few years, I think we have seen how more independent you have become in your way of approaching problems, investigating them, and of course solving them. You work on very interdisciplinary projects, so it requires different work, different various types of knowledge. You did not only work with the data, but also you had to do some lab work and interact with the patients during the PSC study. I would call you a very organized person who likes to work on like one thing at a time, not jumping from one side to another, completely opposite to your supervisor. But I think overall we managed to get very well along. I think that the time of your PhD is not only research, but also finding new friends and different adventures. We know that you are a party boy. I think every conference, it was a great opportunity to meet and to hang out with new and old friends. I remember one of our first conference, which was in Maastricht in 2018, and many of us here remember the evening and the night of the conference dinner, which for many participants, including you, ended up really late with a clear tiredness at the conference the next day, and not mentioning being late to the first session. Nevertheless, this was the conference where you won your poster prize. So overall, it was a great success. The year after was a marathon of the conferences. 3 conferences within 3 months. 3 different countries, Poland, Norway, and White Russia. You presented there your work. You also won another presentation award in Poland. But there was also something funny happened at the Scandinavian Chemometrics Symposium in Norway, where, well, Norway, of course, the main thing we do there, we eat a lot of seafood. I learned that you are against seafood, and you just said like, well, I want to eat meat. The face of the waitress was basically priceless because she said, well, we have seafood or vegetarian, and you were like, no, I want meat. I think a lot has changed in your eating habits since then. I don't know whose influence it is, but that was pretty, nice situation. And I don't know if you remember what we have agreed at the meeting in Scandinavia. Unfortunately, we did not manage to fulfill our promises because COVID-19 pandemic came. And that was, of course, difficult time, right, for everybody. Working at home for you was difficult and was challenging time. But I think it was also the time that made you stronger on very different levels. As I mentioned, you did also some lab work. And I don't know if you remember, I remember it very well, but the first measurements that came out from your lab work and the first QC that we were looking at and seeing such a high variation between the QCs, we just decided like, okay, we all have our weak point, even you. So we just decided like, okay, you will focus on the data and challenging sites there and the multivariate analysis. So, George, I could continue talking, but I think there are people here that also want to congratulate you on this great achievement. Therefore, I would like to congratulate you again, also to your parents, your sister, your girlfriend, Jessica, and they can be all proud of you. May your continued journey be filled with new discoveries, meaningful collaborations, and enduring passion for knowledge. Congratulations.
Thank you, Dr. Smolinska, for a very nice laudation. Dear Dr. Stavropoulos, also on behalf of the Maastricht University, I would like to congratulate you with this certificate and your PhD session. You're a doctor now. You have to celebrate it. I hope you can do that with family and friends. It's an important moment in your scientific career, and I wish you all the best for the future. Congratulations also for your supervisors, of course, Professor van Schoten and Dr. Smolinska, and congratulations for your family and friends here on site and online. And I hope you make it all a great day. I would like to thank the members of the opposition for all their hard work, and I would like also to thank the PEDEL and Luc Peters and Joshua van Veluwe for their assistance, their technical assistance, to make this hybrid session possible. Having said that, I'm almost at a point of closing this meeting. However, I have a few announcements. I would like to ask the audience to leave the room and go already to the rafter where the reception is. Then we will make a photo here with Professor Davis. So I ask the photographer to stay here. And then we are going to take a picture at the foot of the stairs, and then we go to the rafter. And if you think, when can we congratulate the young doctor? That's quite simple. We go first and you follow us. Yes? Having said that, I close this meeting.
So bring the certificates.
Attention. You have to take it.
I know.
Yes.